Thanks, Phil. Thanks for the invitation today. It's been great to catch up with a lot of old friends. The community here is really vibrant, really great. It's been really engaging. Before we begin, I would like to note that I did come with a Halloween costume. I'm dressed today as Phil Romero. So you can see my thesis defense the day after John knows. I'm just kidding. But thanks for the nice introduction. And I'll tell you today about the work that we're doing in the lab that really has to do with understanding how molecules assemble into specific shapes and how those shapes and interactions between amino acid side chains, what have you, govern their form and functions. And the long-term goal of this particular work is to take that perspective in being able to genetically define the composition of proteins, where amino acids lie, where the proteins go within the cell, and be able to exploit that capability to generate mechanoreceptors so that we can program or probe how cells exchange forces between one another, or perhaps someday probe these mechanical signaling events in the body. Okay? So with that said, I'd like to just remind you that when we started this project, we wanted to engineer cells to sense and respond to physical stimuli like mechanical force. Okay? And as a graduate student, you might be thinking, oh, why is this interesting? Why might we want to do this? Well, I'll give you some examples of this mechanical examining of the life processes that we do in everyday life. For instance, you may be at the supermarket, perhaps you're at Whole Foods, Trader Joe's, wherever graduate students are shopping these days. And in order to select the right produce, the avocado that's just perfect for consumption, you might give it a mechanical test. You might grab an avocado and feel how squishy is it. If it's too squishy, it might be too ripe. If it's too firm, it's too green, too early to eat, and you might be looking for the avocado of just the right stiffness. Okay? Now, at the microscopic level, we know that cells in the body are carrying out these mechanical question-asking processes all the time, and the example I like to bring up is the one that follows. This is actually something that came out when I was a graduate student, sitting next door in the office, right next to Phil's. And this is a report that came from Adam Engler when he was a graduate student with Dennis Disher in 2006. And what Adam found as a graduate student was that one can take extracellular matrices from the body, and one can isolate these matrices from various regions of various types of tissues. And if one decellularizes them, removes all the cells, and then grows cells, or stem cells specifically, on these elastic substrates, there's physical information within the mechanical properties of this matrix that can guide mesenchymal stem cells down various fates. So for instance, if you isolate ECM from the brain and you challenge that mesenchymal stem cells with that ECM, they're able to interpret the Young's modulus that's present in that substrate, and that information guides them to become neurons, or for instance, muscle cells, or take on an osteogenic fate, depending on the physical information that is provided within. Okay? Now, in a clinical context, in everyday life, somebody might examine themselves and find that the presence of a lump that might motivate them to go see a doctor and figure out whether the change in stiffness in some region of the body may be an indication of something's wrong, perhaps solid tumors forming. Of course, a physician will have to examine that individual, hopefully do a biochemical biopsy to look at whether a change in the stiffness of various soft tissues here represents something as some malignant or not. Okay? So that's 3 examples from everyday life, whether it's the cellular level, clinical level, or the grocery store level, of how mechanical information can guide decision-making processes. Okay? So with that in mind, we asked, how might we program these types of processes such that cells can be directed to sense some specific mechanical information, and then extract that information, and then carry out a specified biochemical action in return. Okay? And so in a synthetic biology approach, we asked, what types of transducers could we program into cells such that they could detect a specific cue and then now enact a specific response? Okay? And of course, I love proteins. I'm very biased in that I think that proteins are able to carry out everything that we would like to do in biological systems. And of course, nature probably agrees with me as well, because many of the mechanosensitive proteins or mechanotransducers that we know exist in nature actually are themselves proteins. And proteins are, of course, amino acid polymers that fold into unique three-dimensional shapes. But they also can be subjected to force-induced changes in those three-dimensional conformations. So for example, if I take this blue globular protein here and then attach a handle to the N and C terminus of the protein and apply force across the protein, I'd be able to introduce energy that could disrupt the hydrogen bonds, hydrophobic forces that are holding the protein together, and I could stretch the protein into a denatured conformation or into an altered conformation. And along the way, I might generate some conformation that has some signaling outcome or some biochemical output that the cell now can use to interpret the force that's being applied across this protein and then now trigger a subsequent downstream event. Okay? So there are numerous famous examples of mechanosensitive proteins. The two most prominent ones are probably, of course, integrins, but also the piezo ion channels, which won the Nobel Prize a couple of years ago for a discovery of these force-sensitive protein assemblies that live within the plasma membrane and then can exist either in an open or closed state and allow ions to flux through according to the membrane tension that is across the plasma membrane barrier here, like so. Okay? So recognizing that nature already exploits proteins or well-folded amino acid polymers as natural mechanotransducers, we wanted to mimic natural processes and then now apply in a synthetic biology approach a way to engineer proteins to become sensitive to specific forces and then to have that unfolding event provide some biochemical instruction for the cell. Okay? So in our approach, we've been using proteins as mechanotransducing elements. Okay? And so in order to do so, we turn to this protein, which is not traditionally thought of as a mechanical sensitive protein, but mechanical force is required for its activation. And this protein happens to be called Notch, and it's also a protein that really lends itself to synthetic biology applications and rational-based engineering. Okay? So I'll tell you just a little bit about Notch. It gets its name actually from a discovery that occurred about 100 years ago. Famous biologist Thomas Hunt Morgan isolated a mutant Drosophila that he noticed formed a notch within the wings. And it turns out that mutation was located to the gene that encodes for this receptor, and thus we get this name Notch. Okay? And Notch is a highly conserved signaling protein that exists throughout the metazoan family of life, and it serves to inform cells of who their neighbors are. So it allows cells to detect the cells that lie directly adjacent to one given cell. So it allows cells to identify, let's say, their baymates. Okay? And so Notch typically exists at the cell surface, and one can divide Notch into three modular components, and I'll list them out one by one. At the cell surface or at the N-terminus, Notch has this ligand binding domain that allows it to recognize ligands that are typically expressed on the surface of neighboring cells. Okay? And on the intracellular side, in the quiescent form, in the off state of Notch, lies this transcriptional regulatory domain that typically lives at the plasma membrane, but then can move its location within the cell depending on when Notch is activated. Okay? And as I mentioned to you earlier, Notch is a protein that actually requires force for signaling. And this was a debated aspect of Notch biology for many years. There were sort of two camps in the Notch field. One group was really convinced that Notch relied on clustering of receptors in order to mediate signal activation, and others were convinced that mechanical force had to be applied. And the reason why these two hypotheses emerged was due to a very simple observation. So we know that Notch guides numerous developments in vivo, et cetera, but human cells sitting in a cell culture dish can also carry out Notch signaling. And the observation was in the 80s that if you take soluble Notch ligand binding domain, so the protein that recognizes this part of the receptor is usually encoded on an adjacent cell, if you isolate that domain and you apply it to Notch-expressing cells, you can confirm that that domain binds the receptor, but typically that's not enough to activate the signaling pathway. What one has to do is that one has to take that protein and one typically immobilizes it to the surface of a cell culture dish, and that event of immobilization allows the receptor to be activated, and thus sparking the hypothesis that Notch must either require clustering or the application of tension in order to activate the signaling pathway. Now, it turns out, it looks like it's really a mechanical force that is required to activate the receptor, and in the natural case, that force is not delivered via an immobilized protein that one coats to a polystyrene tissue culture treated dish. That force is actually delivered via the endocytosis of a bound ligand by the adjacent cell. So the cell next door will be expressing a ligand, that ligand will interact with this Notch receptor here, and then the active endocytosis applies tension to the receptor, which induces the unraveling of this region that sits in between the ligand binding domain and the transcriptional activator domain. And that region is called the negative regulatory region. Okay? And so that is the mechanical switch that controls the signaling activity of this receptor. So when one applies force against this protein, this region unravels, and what happens actually is that these individual globular, what's called LNR motifs, are displaced away from these blue diamonds as I've drawn it. These blue diamonds, as it turns out, has a space in between because the receptor is actually cleaved by furin convertase in the Golgi. So you have this single polypeptide that's synthesized in the ER. It traffics into the Golgi, where it's recognized by the now famous furin convertase, which clips it, and then now you have two polypeptides that stay together as a covalent heterodimer. That goes to the cell surface, okay? Now, once at the cell surface, it binds the ligand binding domain. Endocytosis actually drags the gray region here and all these green Pac-Men and half of the blue triangle into the adjacent cell. And that event reveals a protease site that was previously hidden in the receptor, and we call it S2 for cleavage site 2. Once that protease cleavage site is revealed, a cell surface protease called ADAM10 can now collide with the receptor, chop it right at this position here, and that cleavage at the S2 position generates something now that is cleaved at an intramembrane position by gamma secretase, and that last cleavage at position S3 results in the liberation of the transcriptional regulatory domain such that it can go into the nucleus and turn on your favorite gene. Okay. This last step right here, as I mentioned, is regulated by gamma secretase. And gamma secretase is this transmembrane multi-subunit complex, and its structure has been solved by cryo-EM in recent years by Yigong Shi and others. And it forms this very elegant assembly that sort of looks like this trash can here with this bulky lid. And the way that gamma secretase works is that its substrate specificity is quite broad. It can cleave about 140 proteins in the proteome so far as we know. But what it looks for are proteins with single transmembrane helices that lack bulky ectodomains. So in the quiescent form of the receptor, this bulky domain, this negative regulatory region of Notch here, prevents the lateral diffusion of this transmembrane region into this trash can, okay? And that lateral diffusion is prevented by this green bulky region here called nicastrin. So you could imagine now Notch in its off state being excluded from the gamma secretase catalytic barrel, yet when cleaved at position S2, you get this transmembrane stub that can readily diffuse in and be chopped up at that intramembrane position. Okay. So, in addition to all those tidbits, really cool feature about Notch, probably my favorite feature, is that it's highly programmable. Folks in this very room, Scott Cole has been involved in redefining the inputs and outputs that can be sensed across this pathway, okay? So I mentioned to you the 3 different regions before that comprises the Notch receptor. But if one were to reprogram this receptor, it's as simple as ordering a DNA sequence now in which the input and output, or the extracellular and intracellular domains, are replaced with almost essentially whatever you want. And this is a powerful approach that's been used in the synthetic biology realm in order to define the signals that cells can sense, or the other cells that a given cell can detect, and to define the outputs that they engage in response to those other cells. Okay? And so this whole process here really involves a protein engineering technique in which the native receptor now is reprogrammed in order to now examine other types of cells. So instead of looking for cells that express Notch ligand, you can express a synthetic Notch receptor or a syn-Notch receptor that looks for instance, a tumor antigen. Okay? Now, but in addition to that, recognizing that although you can engineer syn-Notch to detect different biochemical stimuli, we also recognize that Notch requires this mechanical stimulus for that signal activation. Remember, soluble ligand bound to receptor, natural or synthetic, actually does not lead to activation. One must apply tension to the receptor to reveal that cryptic protease site. So at this stage, we asked, would it be possible to now not only reprogram what the cell senses in terms of its biochemistry, but could we also reprogram the mechanical forces that are required to activate this signaling pathway? Okay. So once again, with our goal of engineering how cells sense force, we asked whether this transducer now can be, in terms of the syn-Notch scaffold, whether this region could be reprogrammed to respond to specified and distinct levels of mechanical tension. Okay, so at the time when we started, single molecule biophysical studies of the natural Notch negative regulatory region were converging upon a value of about 5 piconewtons of tension as a critical threshold for the unraveling of this receptor and the activation of the pathway. And these were based on studies carried out by multiple independent groups, and they all converged on right about this minimal tension value for the activation of the signaling pathway. And if you think about it, 5 piconewtons is actually not very much energy. It's just above thermal fluctuation. It's about, let's see, a couple kBTs. And so we're wondering if Notch represents a minimally sensitive, mechanosensitive protein, if we were to fine-tune the sensitivity of this receptor, which direction might we go now? It didn't seem like going down or tuning down the receptor sensitivity would make any sense. So we asked, how might we increase the sensitivity of this mechanically sensitive signaling protein? And as I mentioned before, when tension is applied to the receptor via the endocytosis of a bound ligand, that endocytic energy displaces these 3 Pac-Men that I'm now designating 1, 2, and 3. So with a very simple logic, we asked whether if we were to add now an additional domain that would to this receptor, so if these 3 LNR domains define the mechanosensitivity of the receptor as 5 piconewtons, could we add a 4th domain to this set of Pac-Men to now elevate the mechanosensitivity of a synthetic Notch signaling protein? Okay? And the rationale here was simple, in that if we added an additional protein, this protein would interact with the NR in a way that would elevate the mechanosensitivity by requiring more force to break those hydrogen bonds, hydrophobic interactions, et cetera, what have you. Okay, so from here on out, I'll tell you about 2 types of proteins that we compare. We compare a standard syn-Notch based signaling protein, and we'll compare proteins in which we contain this strengthened NRR domain, or SNR domain, as I'll abbreviate for strengthened, not a negative regulatory region. Okay. Now, we were lucky because at the outside of this project, there was an existing and known protein that was able to interact with the NRR, for which there was a reported sequence and a high-resolution crystal structure. And the reason why those things exist is because it turns out that in various conditions, prominently T-cell acute lymphoblastic leukemia, in that condition, in this blood cancer, Notch is typically mutated within this mechanosensitive region such that it spontaneously activates without force, or this condition carries with it mutations that destabilize this region of the signaling protein. And folks at Genentech have generated antibodies that are able to recognize human Notch1 and clamp it into its closed conformation, right? To reinforce that auto-inhibited state. And that crystal structure and the sequence information was available in the PDB. So what we did was, in order to make our strengthened NR, we simply extracted that sequence, generated a single chain variable fragment from it, and then integrated that antibody fragment right into the SynNotch scaffold. Okay. We sent the sequence off to IDT. It came back, cloned it into mammalian expression vector. And prior to carrying out further detailed analysis, we wanted to make sure of three things. It's easy to order the DNA, and it's easy to transfect the receptor into cells. But in order for the system to work in the way that we expected or needed to, we wanted to confirm that the thing got to the cell surface. Okay? And we wanted to confirm that the antibody fragment that we've integrated interacted with the protein in the intended intramolecular manner. And we wanted to make sure that the receptor could be activated. So I'll just take you through the set of controls that we did to confirm those three aspects real quick. Okay? To confirm that the receptor gets to the surface, we designed constructs that contained a MYC epitope tag in the receptor, and then we stained cells with fluorescent anti-MYC antibody under live conditions with the rationale that the intact antibody wouldn't be able to permeate into the intracellular parts of the cells. Okay? And in transfected cells, what we found was that when we stained cells for the MYC epitope, cells lit up with green fluorescence, both in the case of NRR expressing cells and SNR expressing cells. Okay? Now, to confirm that our integrated antibody fragment interacted with Notch in a way that would clamp it in the closed conformation, we carried out a second stain. And in the second stain, we used exogenous anti-NR antibody that we purchased. And this antibody is conjugated to now a purple dye. And we stained both cells transfected with the NR-containing construct or the SNR-containing construct. And we asked now whether they would light up in this magenta color. And it turns out cells that express the regular receptor without the integrated antibody fragment were reactive. So wherever we saw green, we saw magenta as well. But cells expressing our engineered construct now, they lit up with green because their MYC epitopes were accessible. However, the antibody now could not bind the negative regulatory region of the receptor because of that genetically integrated interacting domain was occupying that same site. So this gave us some indication that the receptor was folded in the correct intended intramolecularly interacting conformation. Okay. The next thing we did was we also wanted to confirm that we could activate the receptor. After all, we'd fused an inhibitor directly into this critical part of the signaling protein. Okay? And so in order to do so, we carried out an experiment where we took now the synthetic ligand, not the natural Notch ligand, but we took the synthetic ligand that's recognized by the antibody fragment, not the one that's fused and made to bound the NRR, but one that binds, in this case, fluorescein. Okay? And we immobilized fluorescein on the surface of a plate, and then we grew cells that express SynNotch on the surface of that plate. And we asked whether the liberation of the transcriptional effector domain could now turn on some reporter that we could detect. In this case, the translocation of this reporter would go in and turn on the expression of a luciferase that we could measure easily using a chemiluminescent assay. Okay? And so we compared signaling from both our NRR-containing and SNR-containing receptors, and what we found was that in the absence of ligand, we get minimal levels of expression of the reporter gene. In the presence of the immobilized ligand, which remember, the immobilized ligand is required to apply that tension to the Notch receptor to unfold this region here. In the presence of immobilized ligand, we could induce signaling in both the NR and SNR-containing constructs. And furthermore, if we inhibited the proteases that were involved in cleaving S2 and S3, we could knock that activation down. So this told us a few things, that this receptor, when fused to that inhibitory antibody, could be activated, and that it was inactivated in a way that was similar to regular Notch, in that you need a mobilized ligand and cleavage at S2 and S3. Of course, the activation is less, but that's to be expected. We fused it, again, after all, we fused an inhibitor directly into this protein. Okay. So at this stage, though, now that we knew that the protein was localized to the surface, interacted with itself, and could be activated, the next important question was now how much force, or how much more force potentially, was required to activate the SNR-containing construct over the NRR-containing construct. And so in order to probe this question, we borrowed a technique from T.J. Ha's lab. And this technique is called the tension gauge tether assay. And it's a really simple technique, and it works as follows. One takes a synthetic ligand and one immobilizes it to a dish, to the surface of a dish. But instead of immobilizing it passively, what one does is you attach the ligand to the surface of a dish via a bond that can be broken. And in the case of these experiments, we use short double-stranded DNAs that we ordered directly from IDT. And we asked IDT to attach a fluorescein ligand to our receptor because our receptors encoded an anti-fluorescein antibody fragment as their ligand recognition element. And the basis for the technique is that when one applies cells that express receptor to these ligand-coated surfaces, you have now a tug of war between two rupturable bonds. One rupturable bond is the mechanostability of the receptor, so either the NRR or SNR-containing domain right here. And the other rupturable bond is the one that's used to attach the ligand to the surface of the plate. And depending on which one is stronger, you can make an accurate prediction of which entity will win in this tug of war. Okay? So in the case that you have a mechanoreceptor that's able to endure more tension than the pairs of DNAs that are holding the ligand to the plate, you'd expect the receptor to stay intact, and then the DNA strands might be torn apart. And in this case, signaling would not ensue. Now, in the case where the DNA tether that attaches the ligand to the surface plate is really strong, that bond is expected to endure. And in this case, the protein element should lose in this tug of war. And when the protein element loses, you get unfolding, you get a revelation of that S2 site, cleavage by gamma secretase, and then translocation of that transcription factor into the nucleus. And in this case, we turn on, in the experiments I'm about to tell you, instead of turning on a luciferase gene, we turn on a fluorescent protein, HGBM cherry, so a fluorescent histone marker. Okay. And by varying the strength of the DNA bonds that hold the ligand to the surface of the plate, we can now play this tug of war across using DNA pairs of known tension tolerances. So DNA sequences that have been calibrated by single molecule AFM, fully available in the literature. We know exactly how much force is required to break those two DNA strands apart. Okay? And the assay is actually very simple. We just ordered these two DNA strands from IDT. We asked IDT to attach biotin to one strand so that we can immobilize it to a neutravin-encoded dish. And then we asked IDT to attach fluorescein to the other strand, which acts as a ligand that the synthetic receptors can now bind and recognize. Okay? And fluorescein, actually, it turns out the interaction of fluorescein with the antibody fragment that we've used in these experiments have been previously calibrated by AFM as well. So we knew that the interaction between the ligand and the ligand binding to region was not the weakest link in this chain of mechanically labile bonds. Okay. But we challenged cells that were grown in the presence of various types of ligands, including ligands with 12 piconewton rupture sensitivities, 54 piconewton sensitivities, and then at various positions in between. And the strength of these, it turns out actually that these various sequences of DNAs, these are the same sequence composition, but their mechanosensitivities lie actually within the relative orientations of the attachment handle and the ligand binding handle. So for instance, this 12 piconewton tether is easier to break these two strands apart when one applies the receptor at this region here. The cells can now pull on the strand, the red strand with the red bases, breaking those interactions one by one. whereas it's harder to break these two strands apart from one another because the cell would have to break all these bonds simultaneously. So you can think of it as a difference between an unzipping mode and a shearing mode that endows this pair with 12 piconewtons and this one with a 54 piconewton sensitivity. And by varying the position of the biotin immobilization handle everywhere in between, we can get sensitivities of 23 and 43 piconewtons, et cetera. Okay, so in our experiment, because we knew from previous studies that the NRR has a rupture threshold of 5 piconewtons, we expected cells expressing the regular SYN-Notch protein when exposed to these various TGTs. We expected them to all light up with our reporter HTBM cherry here because the receptor, because of course 12 is more than 5 and so on and so forth. But in the case of our strengthened receptor, we were hoping that a set of cells would be silent to some of these TGTs, yet at higher tension values, we could induce some rupture of that protein and get some red nuclei. Okay, so I'll take you through the data step by step. We quantify or we examine the signaling activation in two ways, and we compare cells in two ways using one flow cytometry and two fluorescence microscopy. And in our experiments, we'll compare cells expressing the NR-containing receptor or the engineered SNR-containing receptor. And so in the absence of ligand, we can transfect cells. We know they express the receptor, expose them, grow them in the absence of ligand, and then measure how much leaky activity emerges from these cells. And if we look for HTBM cherry, we see a minimal level of leaky activity for both the NR and SNR-containing receptor. Now, if we grow cells in the presence of those 12 piconewton tethers, we activate the regular SYN-Notch receptor as expected because 12 is more than 5. But we were pleased and a little bit nervous to see that cells that express the SNR receptor didn't respond to 12 piconewtons. Now, when we increased the magnitude of tension that we applied to the receptors, we of course, again, activated the regular SYN-Notch receptor. And now when we applied 54 piconewtons of tension to our SNR constructs, we were getting a little bit of signaling activation, indicating to us that now our receptors, when we hit them with a lot of force, they begin to express a little bit of that reporter gene. And as sort of the upper limit positive control, we simply used immobilized biotin, and we immobilized immobilized fluorescein. We immobilized fluorescein to those neutravin coated plates just by using fluorescein directly conjugated to biotin. And at this level, we get a little bit more gene expression from our strengthened construct. Okay. So since we'd shown now that the thing gets to the surface, interacts with itself in the way we intended, and that we can activate the receptor within the physiologically relevant mechanoactive regime, next question we had was, can we now tune this sensitivity? So we have original receptor that nature's given us, and one that we've engineered by inserting an antibody fragment, and that actually gave us quite a big spread. So if you remember, I told you earlier that the NRR requires about 5 piconewtons of energy in order to activate that signaling pathway. And the engineered construct, it turns out, looked like it needed about 54 piconewtons. So we increased the mechanostability of this protein by about 10 times, and that's a big, big gap. So our next goal was to populate this space in between. And it turns out this is the physiologically relevant regime for single molecule mechanoactive forces. So cells can generate force through things like actin polymerization, and depending on the number of actin filaments that are polymerizing, that are tethered to any given receptor, you can get up to about 54 piconewtons of tension. Okay. So in this last part of the talk here, I'll tell you about the work we did in order to fine-tune the mechanosensitivity of this receptor. And this was all guided by the structural information that's available in the PDB. So sort of inspired, though, by the natural sensitivity of the native NRR, which we sort of thought of as the interface interactions between the regions shown here in dark gray and light gray, or the regions highlighted in yellow. We thought that if the sensitivity of the NRR was defined by this region here, one could think of this yellow shaded region as the mechanoactive site. And we knew from the cancer literature that mutations to this region result in the destabilization of this mechanoactive site, which alters its mechanosensitivity. For our engineered SNR construct, we thought that the mechanoactive site might be between the antibody-antigen interaction, and we were lucky that the detailed information in the PDB gave us a guide from which we could now select individual positions to mutate in order to weaken this interaction, with the hypothesis that through that approach we could fine-tune the sensitivity of our receptors. I won't take you through all the mutations. I'll take you through just a few. The ones that in particular that I chose is because, when I give this lecture to bioengineering audiences, I like to walk them through the biochemistry. Of course, this audience doesn't need any review on that end, but I'll show you positions now that exist between the antibody fragment shown in magenta and the NR components and the NR shown in gray. And I'll draw your attention to this residue in particular, tyrosine 186, which resides on the antibody fragment. It exists within the CDR. Okay? So when we saw this position in the crystal structure, we thought immediately that there were two obvious mutations that we could introduce to position 186 that we hoped would incrementally weaken the receptor such that SNR would now become easier and easier to activate at lower tension values. And so we made those mutations, went ahead and then expressed those receptors in cells, and then carried out the same type of analysis that I told you earlier using those TGTs. So I'll compare now cells that express the SNR-containing construct, and in the wild type sequence, in the synthetic wild type sequence, they contain tyrosine at position 186. And I'll compare their signaling intensities via flow cytometry, quantifying that mCherry reporter gene to a receptor now in which this tyrosine has been mutated to phenylalanine, or in other words, we've eliminated this paraphenol group here, and in the native context, the two hydrogen bonds that are formed between the tyrosine's OH moiety here and backbone elements are ablated. Okay? So we can now probe specifically the mechanostabilizing contribution of this pair of hydrogen bonds. Okay, so when we have cells that express SNR versus this mutant, we can challenge them with 100 piconewtons of tension, our upper limit control, and we can see we max out the signaling activity of the reporter, and we get signaling extents to the same degrees. Now, when we looked at the 12 piconewton tension gauge tether, consistent with the data from the previous slide, we saw that 12 piconewtons wasn't enough to activate signaling of the SNR-containing construct. Now, cells that express the mutated version, in which we remove this pair of hydrogen bonds, when we expose them to 12 piconewton TGT, we got a little bit more mCherry expression. And what this told us was that this receptor was a little bit mechanically destabilized, or it could detect, to a small extent, the availability of a 12 piconewton tension-based stimulus. Now, when we looked at the other TGTs, the higher values, let's see, at 43 and 54 piconewtons, we saw that the same trend continued. Okay, so in the case of the SNR construct, the 43 piconewton tension gauge tether could activate the receptor to an extent, and that the same tether, so the same amount of tension, created even higher levels of mCherry expression in this mutated version. Okay, so if you could think of this what we've done here as we fine-tuned the receptor by eliminating this pair of hydrogen bonds, and now just by eliminating this OH group, replacing it with a phenylalanine, we've engineered cells that can detect 12 piconewton tensions, but can barely tell the difference between 43 and 54 piconewton stimuli. Okay. Now, that's nice and everything. The next thing we wanted to examine, though, was whether we really knew what we were doing. So instead of just eliminating the paraphenol group here and adding a phenylalanine at this position, we next asked what would happen if we introduce alanine at position 186 with the rationale that that mutation could be used to probe the mechanostabilizing role of this aromatic ring. Okay? So I'll compare now data that we collected for the 186F mutant and the 186A mutant. As you can see here, when we eliminate both the hydrogen bonds and the ring, we have a receptor that's really trigger happy. So 12 piconewtons of tension is able to activate the receptor to a greater extent than the 186F mutation. Okay. Now, using that same rationale, we created a family of about a dozen or more various mechanoreceptors with sensitivities that span the range between 12 and 54 piconewtons. And the next thing we did was, now that we'd shown that we could engineer the proteins in different ways to have different sensitivities, we asked whether we could use these proteins in ways that mimic natural processes in the body. So I told you earlier about this paper that came out when I was in graduate school back in the dark ages, in which stem cells were found to be able to detect the physical properties of the ECM and make decisions according to that physical information. Okay? In a synthetic biology approach, we carried out an application in which we asked whether we can now use our mechanoreceptors or combinations of our mechanoreceptors to mimic this decision-making processes, in which we can take stem cells and then guide their differentiation into these various different fates according to the mechanical stimuli that we apply to the Notch or SynNotch receptor. Okay. And our first approach was a more simplistic approach than the one that nature carries out, of course, but we simply had a threshold now where we could define the receptor sensitivity or the amount of force that we're applied to cells in order to induce differentiation of fibroblast cells into a muscle cell-like state. And it's a very simple process. We simply take fibroblasts and we express these receptors in this type of cell. And we borrow the SynNotch approach to now vary the intracellular domain that's encoded within this receptor. And in this case, activation of the signaling protein releases this transcription factor that turns on a synthetic version of MyoD. And MyoD is this natural protein that, when expressed in fibroblasts, it's a master regulator, a transcriptional regulator that guides the conversion of fibroblasts into a myoblast-like state. Okay? And so we expose cells now to various tensions, so TGTs of different values, and we compare two types of cells that contain the weak mechanoreceptor, the NRR-containing receptor, or cells that contain that strengthened NRR. Okay? And so in the absence of any ligand, when cells expressing this receptor, we see that the signaling pathway is off, and the cells, they stay as fibroblasts. And we know that because we know cells are there. We've stained the nuclei with DAPI. And in order to determine whether any differentiation has occurred, we stain the fibroblasts or the myoblasts with this marker here that indicates us it's myosin heavy chain, which is expressed only in the muscle cell-like state. Okay? Now, when we expose cells that express the regular receptor, the NR-containing receptor, to 12 or 54 piconewtons of stimulus, we can now drive that conversion of the fibroblasts into the myoblast-like state. They start to express myosin heavy chain, and then they become fused and multinucleated, so we know that they become muscle-like cells. Now, cells like a cell line that we generated and carried out and analyzed in parallel with this line are cells that express essentially the same components, with the only difference being that they contain that strengthened mechanoreceptor domain instead of the natural NRR. In the absence of ligand, they stay as muscle-like cells. And of course, as expected, 12 piconewtons of tension is not enough to activate the signaling pathway, so they again stay like muscle-like cells. But in their old age, if they put a little bit more time in at the gym, you get something that sort of looks like muscle cells. But again, we can activate now the signaling pathway into an elevated level of tension to generate something that looks like the myoblast-like state. And that's just a zoomed up view. Okay. Last thing I'll probably tell you about is how we are trying to get one step closer to mimicking the natural processes. So I'll go back to this example where the finding that mesenchymal stem cells can become these various states, and it turns out that they're able to interpret or distinguish between soft, medium, and stiff extracellular matrices, and then take on this myogenic fate, similar to the fibroblast cells I showed you in the previous slides, but they do so only in response to medium stiffnesses. Okay? So you can think of these stem cells as sort of like Goldilocks, in the trespassing in the bear's property and eating the various porridge, and the muscle cell like fates of the stem cell occurs only in response to the avocado of the right stiffness. Okay? So how might we mimic this process? Well, again, we turn to synthetic biology strategies, and in this case, what Chris Sloas, my student, implemented was what the synthetic biologists call the incoherent feed-forward loop, in which now we are combining two receptors of distinct mechanosensitivities, and we combine their two actions together to get signaling only at an intermediate level of tension, so that the cells stay as fibroblasts or have a gene that remains in the off state in response to low tensions or high tensions, and that the signaling pathway is activated at only levels that in between. Okay? So for instance, can we guide, the question we're asking is, can we guide cells now to pick the avocado of the perfect ripeness for tonight's guacamole? Okay. And in order to construct this incoherent feed-forward loop, what my student did was, instead of expressing one receptor inside these fibroblasts or inside reporter cells, we now express two receptors together as a pair. And these receptors contain the same extracellular ligand binding domain. They contain distinct mechanosensitive domains. So we look at a weak receptor and a strong receptor. We express them in the same cell together. And then we vary their intracellular domains. Okay? The weak receptor contains a GAL4 base intracellular domain. And when that's released by gamma secretase, it goes on and it turns on a green gene within that's encoded within the nucleus. And the strong receptor now contains a distinct intracellular domain that turns on the expression of a red fluorescent protein. And accompanied with that red fluorescent protein is we've hooked up the expression of an inhibitor. So something that can go on when activated and turn off the expression of this first arm of the circuit. And our rationale was that we can now expose these cells to varying levels of tension. In the absence of tension, we expected both arms of the circuit to be off. But at some low level of tension, we might activate one receptor but not the other. In fact, we expected that from our calibration assays. And in this case, we'd expect to see the green gene on, but this red gene off due to the sub-threshold activation tension that we've applied to cells. But if we were to bump up the amount of stimulus that we applied, we could now engage the second arm of the loop, which when activated would automatically turn off that first arm. Okay? So you could think of this green, the expression of this green gene here as the response to the stiffness values between two windows of tension. And that is indeed what we see. So if we look at the reporter genes, and this is in fibroblasts, but just in HEC293 cells, in the absence of ligand, both reporters are off. When we stimulate cells with a 12 piconewton stimulus, we can engage that first arm of the loop and express M cerulean or a CFP-like gene. And then when we activate that second arm of the stimulus, we turn on that red fluorescent gene, which is accompanied by the transcription factor that silences that that first gene, the CFP gene. Okay? So we think now we can combine receptors in different ways and install various decision-making logics into engineered mammalian cells. I might have time for the last and favorite part of the story. I'll try to go very quick. The last part of the story has to do with applying these tools, now not for synthetic biology applications, but using them in a way that could teach us about natural mechanisms of life. Okay? So I told you earlier that the mechanosensitivity of Notch receptor is about 5 piconewtons. People figured that out by purifying proteins and applying magnetic beads and single molecule forces against purified protein, but also in cells. And that's well established. But knowing that the receptor has this 5 piconewton threshold sensitivity, we asked, could we use our system now to measure the amount of force that ligands actually apply to the mechanosensitive receptor? So if 5 piconewtons breaks this thing, how much force is it that the ligand in the natural context or in synthetic construct, how much force is being applied by the signal sending cell? Okay? And it turns out the force delivering entity that activates notch signaling at a cell-cell interface, as I mentioned, is endocytosis. But that endocytosis is regulated by the intracellular domain of a ligand protein. The intracellular domain actually becomes ubiquitinated, and that ubiquitin recruits adapters that link the ligand, the transmembrane ligand, to epsin, clathrin, and polymerizing actin filaments. And that set of proteins together drives the internalization of this ligand along with the bound notch receptor, and that's the force delivering event that stimulates the signaling between these two cells. Okay? So knowing that ligands that contain the notch intracellular domain sequence are able to apply force to bound receptors, we now use the synthetic-based sequence in which GFP is introduced as an extracellular element, and we express GFP as an artificial ligand fused to the notch's natural intracellular domain, and we ask the question of how much force now can this synthetic protein apply to a notch-expressing cell when it becomes ubiquitinated, bound by epsin, et cetera, so on and so forth. And to do so, we simply generated ligand-expressing cells and then challenged them with our receptor-expressing cells using the NRR-containing receptor, the strengthened SNR receptor, and the various mutants in between. And we simply asked, when does signaling occur and when does it stop? With the rationale that the amount of force that is applied by this ligand is limited. It's tuned, of course, to the natural context, and we wanted to know if it could now activate SNR or any of our various mutants. Okay. And so when we mix those synthetic ligand-expressing cells with cells that express our various receptor constructs, we use 2 ways, again, to visualize the activation of the signaling pathway, relying on the expression of H2BM cherry. And we found that the ligand was able to activate the syn-notch construct. This artificial ligand was not able, however, to activate signaling of our strengthened receptor. And this is the strongest version of the receptor. But when we compared the signaling between cells using our weakened receptor variants, we found that the weakest receptors were able to undergo that force-induced ICD liberation, as I mentioned before. And we could quantify these values using flow cytometry. And it turns out that all of the order of these receptors line up exactly as we found that they did in our TGT analyses. Okay. And the last experiment I'll tell you about was a control experiment in which we generated cells that express the ligand now and contains an intracellular domain in which that recognition by the ubiquitin ligase now has been mutated. And so in this ligand, we expected it would get to the surface, it would be able to engage with the receptors, but because it cannot be ubiquitinated, it would not be internalized, and force could not be applied across cells. And here's what we see. Okay, so cells that express the ligand with the delta-like 1, which is one of the canonical notch ligands. This is the active version, and we get this induction of M. cherry. Now, when we express cells that contain the mutated ligand that cannot be ubiquitinated, we knock down the signaling of the receiver cell in nearly all of the cases. So this was real evidence to us that we generated a genuine bona fide mechanoreceptor, synthetic mechanoreceptor, that could now sense the forces that were delivered to a receiver cell by a cell that expresses a ligand. Okay, so I'll just end here by summarizing what we did. My student, Chris Sloas, one of my first students when we started the lab, set out with the goal of engineering how cells sense and respond to force. We applied natural mechanism and exploited the modularity of the syn-notch scaffold to generate a family of receptors, used structure-guided mutagenesis to destabilize those receptors, and populate the space in between. And then we showed that these receptors could be used to activate gene expression responses in response to extracellular attentional cues. And we applied the receptor to investigate the force-delivering values of natural signaling components. And I'll end with thanking the folks who did the work. So the work was started by Chris Sloas, who's gone on now to work at Charisma Therapeutics in Philadelphia, engineering macrophages for cell therapy. The cell-cell signaling work was actually carried out by two students who are still in the lab, Jeremy Tran and Alex Marzilli. They're 4th and 5th years, and if you're ever in Boston, please come by and visit them. They're going to be there for the next 12.5 years as they finish up the rest of their theses. I'm just kidding, but maybe I'm not. We'll see. But together with the rest of the lab, we've built a tool development group that's exploring various areas of cell biology. Yeah. Thanks very much.
Questions?
Great talk. I love the design of all these receptors. I was curious if we didn't get about the sort of kinetics of the turn on across receptors. Because like you have a transcriptional readout, so some of them, the ones that are hard require more force activity, take longer.
Yeah, so my answer to that is a little bit complex because in this case we're using in the cell-cell-based signaling assay, we use cells that express a GFP ligand and an anti-GFP antibody, and that bond itself is actually rupturable. And what we think is happening, the answer to your question is that yes, the stronger receptors do take a longer time to turn on. And the reason for that, we think, is that the stability of the GFP, anti-GFP interaction is just marginally stronger than the strengthened NR interaction. So what happens is that instead of undergoing this yes or no tug of war, the cells are engaging with one another, and the GFP ligand interactions sometimes break, but a fraction of the time, the strengthened NR domains break. And then and that causes lower gene expression levels at our endpoint analysis at 24 hours. And so the differences that we see on flow or that we've plotted by those traces correspond to changes in the rate of gene activation. So thanks. Yeah.
Question way in the back.
So how does the system kind of get reset after it gets cut off?
Yeah, so it is a one and done pathway. So once you cleave the receptor, it liberates it and goes into the nucleus, and then you get this defined window of signaling that corresponds to the number of copies of the ICD, the intracellular domain, that is made into the nucleus, and its half-life in the nucleus. And we use regular Gal4 artificial transcription factors, which turn out to have short half-lives when released into the nucleus. But natural notch actually controls the half-life of the ICD when it's in the nucleus. So there's this whole interesting body of literature about how notch has evolved itself to become this signaling system that's able to quantify the ligand expression on its neighbor and activate a defined gene expression program within a window of time. Now, that was a very long-winded answer to your very direct and good question, but we have another way of controlling also the window of time during which the signaling occurs, and that is to make the interaction between the ligand and receptor cell drug sensitive. So then the signaling pathway occurs only when we have a drug present in the medium. And in this case, we use something that contains fluorescein. And then as a ligand, we use antibody fragment that recognizes biotin. And so this is one of these bifunctional molecules, has two heads, can bind different antibody fragments. And it turns out when we use that molecule, we get very sensitive signaling. So we're able to turn on the communication between these cells at very low amounts of drug.
So two sort of related questions. One is, you have these transfer function curves for different force values. A, what is it? What does the real data look like for cooperativity? And B, how would you change the cooperativity of the system?
We don't know at the single molecule level because our readout is simply gene expression levels that we measure 24 hours later. But if we and it's a slow process. I think the question is, what is it that the
Does it go like this, or does it kind of go like this?
Yeah.
Is what I'm trying to-
There's a whole debate in the notch field, or there's a question in the notch field as to whether those green Pac-Men are displaced by force one at a time or all simultaneously. Now, the single molecule techniques haven't reached the level of resolution yet to be able to record those events as they're applied with loading rates that are relevant to cell physiology. So with regard to natural notch, the question is unknown. With regard to our system, what we think is happening is that we are generating receptors, the sensitivity of which is defined primarily by the antigen-antibody interaction. And so that we think that there's limited cooperativity because as soon as you rip that antibody off the antigen, the rest of the green Pac-Man just fly away as well. So that's the primary gate that we've introduced in the system here.
Any other questions?
This may be a very interesting system, by the way.
Thank you.
This may be a bit of a crazy question, but in the initial system where fluorescein is attached on the surface, and you have your cell, I wonder if there's an effect where expressing many copies of your reporter could cause the cell to migrate less. Because I assume activation is due to the cell wanting to move somewhere,
Yeah.
and if you have just one copy of your reporter, it just easily breaks. But having like a billion of them might cause the cell to just not move at all. And
Right.
Does anything like this happen?
Right. So so we've introduced a avidity effect that allows the cells to stick. That's a great question. It's interesting because the natural ligand receptor interaction is actually a very low affinity bond that undergoes, it strengthens under force. And so, the answer is we probably are seeing those effects in our synthetic system. But I think a more interesting question is to ask how nature has evolved a natural signaling protein that bypasses that. So I have a lot of things trying to synthesize a succinct answer here. And I guess the simplest one is that we don't know, but we would be very interested in knowing. Thank you.
Okay, let's thank our speaker one more time.
Thanks.
That's a good one. So
Thanks.
what's stopping us from just stringing together a bunch of interacting pairs of domains and turning really anything into a